Hello, everyone. My name's Jonathan. Thanks so much for attending today's webinar. I'm a PhD student in John Bowman's lab, currently writing up my thesis, hoping to submit soon. And today I'd like to talk to you guys about some of the work that's been done in this project, which revolves around spores, the genetic development and regulation of spores. And when talking about the context of land plant evolution, spores are perhaps one of the most critical unifying traits among land plants because the presence of fossil spores is the major line of evidence that tells us land plants emerge around 470 million years ago, whereas the first plant macrofossil only appears around 65 million years after that. And to bring this back to present day, these are some of the spores and pollen grains of people who work in the center, and they are model plants. And you can see that there are huge taxonomic conservations in terms of spore and pollen morphology. For instance, rice and sorghum have very similar looking pollen grains. Similar as a lot of pine have pollen grains that look like this. They've got two little sacs filled with air that aid in wind dispersal. And the production of spores are present in every single land plant lineage. And what makes a spore is basically their outer coat. Their reticulate spore coat is made out of a polymer called sporopollenin. And sporopollenin is said to be one of the most durable biopolymers made in nature. And it's because of this recalcitrance that throughout a century worth of research, we still don't know what the molecular structure of sporopollenin is because of its inability to dissolve in a lot of the common solvents that chemistry uses to study molecules. Sporopollenin is said to be a pre-adaptation from the Caryophycean algae. In the Caryophycean algae, cells that are about to undergo meiosis sometimes undergo dormancy and they put up this wall made out of a sporopollenin-like polymer. And this wall is very different from the ones in spores, land plant spores. It's a lot thinner and it's a lot less intricate. But in land plants, we see a lot of diversity in spore shape and ornamentation. And although we do not know what the exact chemical composition of the sporopollenin biopolymer is, a lot of what we know about sporopollenin comes from genetics. This is a putative pathway of sporopollenin biosynthesis in Arabidopsis. It happens in the tapetum, which surrounds developing pollen grains. And we've got a bunch of transcription factors. A lot of them are bHLH transcription factors and MIB transcription factors that activate downstream biosynthesis genes. And I say putative pathway because their positions in this pathway are all figured out through in vitro enzymatic assays. But there are still a lot of question marks. We don't really know exactly where these enzymes fit into sporopollen biosynthesis. The most confident aspect of sporopollen biosynthesis that we are in is in relation to these enzymes right here. We are very confident that these enzymes synthesize polyhydroxylated polyketides, and they make up the backbone of sporopollenin. And it's just a fatty acid derivative. And when we look at the genomes of all extant land plants, we see very strong conservation of a lot of these enzymes and transcription factors. In the land plants that we study, Marchantia polymorpha, transcriptomic data as well as sequence similarity wise, we are able to identify a lot of these transcription factors as well as biosynthesis genes. And one of the very first genes we looked at in Marchantia polymorpha was bHLH37. It's a subclass 2 bHLH that's putative roles is in regulating sporopollenin. And in Marchantia polymorpha, the sporophyte, this thing right here, is very different from the one you see in Arabidopsis. And the Marchantia polymorpha sporophyte, which is these yellow little sacs of spores, has a very simple body plan. It's got a single layer of cells encapsulating the spores called the capsule wall. It's got these really long helical thickenings called the elaters, these long cells that work in spore dispersal. And of course, you've got the spores. And one thing to note about the Marchantia polymorpha sporophyte is that all three of these cell types make their own secondary cell wall modifications. You've got these bands around the capsule wall called annular rings. You've got the helical shaped thickenings on the elaters. And of course, the spores have their sporopollenin wall. And the difference in liverwort sporophytes and the ones of vascular plants is in the fact that for the longest time, we thought that liverworts do not have a tapetum. In Arabidopsis, you see, pollen grains are all surrounded by this single cell layer called the tapetum. And the tapetum is the one that produces all the enzymes and precursors for wall formation in pollen. The same goes for mosses. We thought for the longest time that liverworts make their own, the spores make their own wall. So when we first looked at bHLH37, this would the fact that spores make their own wall would expect bHLH37 to be expressed in the spores themselves. But this was not the case when we looked at bHLH37. Expression was specific to the capsule wall as well as the elaters. And when you knock out bHLH37, what you see is a total absence of a spore wall. Sporophyte growth in Marchantia polymorpha takes around 30 days. Around 15 days, spore mother cells start to undergo meiosis, producing 4 cells in the tetrad. And it is during this stage where spore pollen is deposited and the spore wall starts to develop. And around 25 days of growth, they start to disassociate and become free spores. In bHLH37, all the spores at maturity are still trapped in tetrad formation. And the spore exine, the thick black lines, never form, meaning sporopollenin is completely ablated in these mutants. And this study was published 2 years ago. And since then, we've looked at the transcriptome of these bHLH37 mutants, and we've additionally identified putative candidates for every single gene in the polyketide pathway of sporopollenin biosynthesis. So today I'd like to share with you guys new data on 3 of these enzymes. We've got ACOS5, which is a CoA synthetase. It adds CoA groups to fatty acids. And PKS, which is a polyketide synthase. It turns CoA fatty acids into polyketides. And the products of this pathway is supposedly exported by ABCG transporters or ABCG12. So we can start by looking at the expression of these genes. And so far, I've got reporter lines for ACOS5 only. And in ACOS5, expression is specific to the elaters, and specifically during the tetrad stage at 20 days post-fertilization. And this is when we think sporopollenin is produced at the highest amounts. At 25 days, spores have already disassociated from their tetrads into free spores, and we see that expression in the elaters is very mild. And while I'm still working on reporters for ABCG12 and PKS1, we have single cell data from a related liverwort called plagioclasma. And in plagioclasma, single cell data of the sporophyte shows that ACOS5, ABCG12, and PKS1 are all co-expressed in the same cell classes. So presumably they're very likely expressed in the elaters as well. Well, we'll just have to find out to see later on. What we can do is look at knockout mutants of these genes. And in the Bowman lab, a lot of our Marchantia polymorpha mutants are made with CRISPR. So we just design guides targeting the gene across conserved protein domains. And we isolate full knockout mutations like these, and we look at how they affect sporophyte development. And looking at all three knockouts of these genes, just looking at the sporophyte, the most obvious defect initially is a coloration defect. In wild-type sporophyte development, the yellowing of sporophytes happens at the very last stages of development. Throughout the last 5 days of development, spores turn from green to yellow very quickly. And we see that in all of the mutants of polyketide biosynthesis, this pigmentation is not deposited, it's all accumulated. And we see in the wild type, pigmentation occurs across every single cell type and their secondary cell wall modifications, the capsule wall, the elaters, as well as the spores. But if we look at these cell types across all three of these mutants, pigmentation isn't really picked up. Pigmentation is all accumulated within cells of the capsule wall and sometimes right on the outside of the capsule wall in big deposits. And looking at ABCG12, we see some additional phenotypes whereby the elaters are completely absent of secondary cell wall modifications. They do not put out helical thickenings. Additionally, spores of ABCG12 are also, like bHLH 37 mutants, are all trapped in permanent tetrads within a spore mother cell wall that does not deteriorate. And across ACOS5 and PKS1 mutants, the phenotypes are very similar in that the capsule wall and elaters have a coloration defect, but the spores definitely have an ornamentation defect across their spore walls in both patterning as well as coloration. And we can look at these cell walls at higher magnification through TEMs and SEMs. In the wild type, this is what spores look like under the SEM. And when you slice it open through TEMs, you see that the spore wall, the very densely colored layer, is called the exine. There are two layers of the exine, exine 1, which is very patterned, and exine 2, which is a very even layer right beneath the exine 1. And both exine 1 and exine 2 are thought to consist of sporopollenin. When we look at ABCG12, it becomes very obvious that the tetrad is all still enclosed by their spore mother cell wall. Now, this small mother cell wall usually appears during meiosis, and this integrates during the free spore stage, and this doesn't happen in ABCG12 mutants. In fact, none of the exine layers form in this mutant. And if, in fact, ABCG12 is not related to sporopollen biosynthesis, we would expect to see a lot of sporopollen deposits in the locular space, but that is not the case. For instance, when we look at ACOS5 and PKS1, where we are guessing that sporopollen biosynthesis is somewhat impaired, we see that both exine 1 and exine 2 are severely affected. Exine 2 is completely missing. Exine 1 is loosely deposited all around the spore. The same goes for PKS1 mutants, a very loosely deposited and defective exine 1, and exine 2 is completely missing. And when we look at the interlocular space, we see that deposits of defective exine material is all around the locular space. And because there's such a severe exine phenotype, we can test the integrity of sporopollenin through acetylysis. Acetylysis is the potentially the only assay we have to test for presence of sporopollenin. You basically boil plant material in a mixture of acetic anhydride and sulfuric acid for 4 minutes, and the only thing that should remain is sporopollenin. So this is what it looks like in the wild type. But across all of these mutants, they fail to tolerate acetylysis, meaning sporopollenin is not present in any of these mutants. And because there's a coloration defect across their annular rings and helical thickenings, we can also look at these cell walls in the elaters and capsule wall. And ABCG12, it's very clear that none of the secondary cell wall modifications are happening. And interestingly, even though through light microscopy, the annular rings look somewhat normal aside from the color, we see that they are a lot thinner relative to the wild type. And they're a lot less electron dense. There are white dots, grainy, all throughout the annular rings. Whereas for ACOS5 and PKS1, TEMs don't reveal much about their differences. They look very similar to the wild type in terms of electron density. So there should ideally be a light microscopy based assay to test just how defective these secondary cell wall modifications are in these two mutants. But taken all these results, we see that ABCG12, ACOS5, and PKS1 mutants are very similar. ABCG12 does have additional phenotypes, such as the persistence of the spore mother cell wall, but despite severe spore wall features, they're all fully viable if you grow them up fresh. These spores are severely intolerant to desiccation. If you dry them out for just a week, none of them are able to germinate, which again points to a very, very defective spore wall. And all these results can tell us a few important takeaways about liverwort and land plant evolution. Firstly, the liverwort capsule wall is the one producing the yellow pigments required by all the cell types in the sporophyte. Additionally, the elaters are making up the sporopollenin-based polyhydroxylated polyketides. And these two cell types together make up, they act as a liverwort tapetum, producing spore wall components for the spores. They have different functions, but they both contribute to the spore wall. And secondly, enzymes of the polyketide pathway, such as ACOS5 and PKS1, have been co-opted to the helical thickenings as well as the capsule wall, because we see coloration defects in the mutants. So this co-option to their secondary cell wall modifications likely happened a long time ago, early on in liverwort evolution, because helical thickenings in elaters are conserved across liverwort lineages. So finally, we land at the liverwort pathway of sporogenesis and sporopollen production, whereby the genes that we have studied so far is we know subclass 2 and subclass 3AC bHLHs act together to regulate downstream biosynthesis genes such as ACOS5 and PKS1 within the elaters, and they are likely exported by ABCG12. And it's a very exciting time in sporopollenin research, because we're finally getting to a point where the genetics is starting to link up with the biochemical studies, and we're finally starting to see patterns and know core truths about the sporopollenin ultrastructure, and as well as its biochemical composition. And that's all I have today. I'd like to thank all of my lab mates who I miss dearly, because I've just been at home writing my thesis for the past month. And thank all of you for listening.